2011 paper mentions the need to look at variations in protein coding regulatory as well as structures. Now it says the term causal variance. When you do a population, when you do NGS studies, right? When you do NGS studies, more often than not, what you find is that you will see that you will get maybe 100 different SNPs, yeah? And perhaps maybe 50 something is actually linked and so on. But you're not yet able to pinpoint which are the ones that really matter. Yeah. So this paper says when you look at variation, you need to differentiate, or you need to know what you're looking at. Either you're looking at protein coding, then that one you're talking about amino acid changes and so on. Are you looking at regulatory, or are you just looking at structural? What are structural sequences? Structural sequences are, for example, the centromeric, telomeric. So these are structural sequence. And then at the moment, a lot of studies are just looking at association between phenotype and variation, but they don't really know what the variation does. So this is where current studies are moving towards. I know this variation, let's say for example in a particular region, if you have an A, you have a higher chance of getting disease. If you have B, then you probably not. But now they're looking at why if you get A, you are at a higher risk of getting disease. So this is called the functional aspects of this kind of study, right? So to actually find the causal variance. There's another paper here, number 9, looks at replicability of achieving the same objective across different studies in various populations, and then they compared European studies against Africans as well as patients with familial cases. Now this is, I think, looking at can't remember what disorder, right? But basically they're comparing results from, and they found that when they compare all studies, they said there are 30 SNPs that are important, and of that, in the 30 European SNPs, only 19 is actually applicable in Africans, and of that, only 10 are actually applicable to familial cases. If you're in a hospital, when you get patients with any particular cancer, for example, there are patients who do not have any family history. I'm not saying that the patients do not have family. It's just that in the family there is no history of that particular disorder, and there are some that oh, I have cancer and my auntie, my uncle has it, my late grandfather. So that's a familial history. So so they categorize these two: one who does not have any familial link, and then the other one who has. And then they try to see whether the SNPs are associated with one type or the other. So it says, it says 10 are actually associated with a familial type. Okay. And there is a focus towards 8q24. And from all these different studies, a lot of studies are pinpointing that particular area being most implicated, probably stating that this is one of the more common causes of what's the disorder? Never mind, I'll look at it later. This is paper number 9. Okay. 10 focuses on selected genes to see which. So again, this is a candidate gene basis. That means you have a group of genes, and then you pinpoint that. One important conclusion was that there was no similar level of gene expression in different populations. Why is this important? Highlights the fact that if you do this kind of studies, you need to define your population. You also need to look at the factors that are playing in that particular population. I'm talking about maybe diet, maybe a simple factor like if you ask me, the Caucasians are more health conscious. compared to the Asians. We eat a lot, and although in Americans, for example, there is high obesity. But you just have to look at different. But it depends on what they eat, what we eat in our diet. Even among the cultures within ourselves, within the Malay, Chinese, and Indian, you have different eating habits, different kind of food, different environment. So this is why the conclusion is that when you compare, you won't be able to see a similar expression between different populations. So that's why a population-based study is important, right? NAFLD, this is liver disease. Okay, now this one says you look at variations in the genes, genetics, but epigenetic is also important. All right. I'm sure you know what epigenetics is, right? So in this case, they look at microRNA, DNA methylation, histone modification, ubiquitination, and so on. So different. So it's just saying that don't just look at genetics, because you'll find. I mean, we know genetics. Our phenotype is determined by our genetics plus our environment, and epigenetic is a lot is associated with the environment. When you're talking about environment, it's not just our environment; it's our also cellular environment. All right, so this is what we're talking about. Paper number 12 looks at GWAS and obesity, right? Obesity and adiposity. What is adiposity? And the adipose tissue is where a lot of fat tissues are being stored, right? And this one is a candidate gene study. They look at 7 different genes, and now they are also trying to link variations with function. Note, nowadays scientists have started to question what does this is what I'm just trying to say. Scientists have started to question, and this is what I'm reading at. This is what I'm reading. What do these variations mean? So you're not just saying, oh, this particular variant is linked to that and so on. They want to know now, okay, if it is linked, why? They're looking at fine. They want to understand what and how these variants affect the function. So it's not. That's why now that's why the focus sometimes is look at genes, because any variation within the genes are easily interpreted. Unfortunately, if most they will find later, they will show later that most of the links are not within genes. Another study, genomics of elite. When we talk about genomics, you're talking about NGS, right? Genomic genetics. When you're talking about individual genes, genomics and whole genome. So they're looking at elite sporting performance. What are elite sports? Please don't be confused. Elite sport doesn't mean that it's for the elite group. No, elite sport is just competitive sports. You had the mass sports where you play football just for the, but competition based, these are elite sports, elite sporting performance. They're looking at genetic variation that predisposes. What does predispose means? You have the tendency to, right? Predisposition, injury predisposition, whether you are easily injured, response to training, how easy your muscles develop, sprint performance. Here, when you look at the muscle, there are 2 types of muscle fibers. You have the slow, quick twitching, and then the slow twitching. I can't remember when I was in Sukan. I actually like reading this. You see, the fast twitching. If your muscles can contract very quickly, you are suitable for sprinting, short distance running. But if your muscles are the slow type, that means you have more endurance. So you tend to fare better in cross country, long distance running. you won't be good at sprinting. Yeah, so they look at genes that determine this, and they found some correlations. So this is, again, genomics of elite sporting. This one is also on sports, and then they look at submaximal exercise heart rate, whether your heart, that's also important. If you are active, if you are fit, then your heart rate will be very slow, right? Relax. And that also has got some genetic components in it, linked to 9 SNPs. Emerging role, so that one is 2013. Yeah, there's another one, paper later that I think in 2015 that actually looks at sporting genes as well. Now, emerging role of epigenetics in IBD, irritable bowel syndrome. They've looked at 163. They found 160 susceptible locus. So that means these are genetic components. 163 locus that are actually linked to, that determines whether you have IBD or not. But still some, and yet is still some patients that does not show this link. So what does this mean? So does not show link to what is already known. Sorry, there's a spelling mistake there. And this is because genetic forms only a portion of overall disease variant. So again, phenotype is genotype plus environment. Must take into account gene-environment interaction, and this gene-environment interaction is mediated by epigenetics. What are epigenetics? Histone expression here, it says CPG islands. You know what CPG islands are, right? CG, 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 CG. The occurrence of CG in particular regions. Methylation, because these are hotspots for methylation. Methylation is whether they're methylated or not. Histone variation, whether the DNA is compact or not. MicroRNA, among others. Right, so this says you need to look at all this, not just look at the genes, they said. 16, GWAS and suicidal behavior. Initially, they look at 8 SNPs, and then they find, and there are 15 SNPs. And then they review all papers that talk about suicide and found similarities between studies, and this is what they found. These are the conclusions. Summary effect sizes were small. What is the effect size? Effect size is a statistical feature that says, okay, something like the effect size, if it's small, that means, yes, it has an effect, but it's not very significant. If it has a big effect size, that means it's very important. Yeah, so that's effect size. So summary effect size was small and rarely statistically is it significant, but maybe it contributes a little. Heterogeneity between studies were often, heterogeneity means between study, this person found different results from the other. Right? Heterogeneity, that means in one population is caused by different genetic factors, other populations are caused by different genetic factors. There were no time trend. Yeah, sure.
Yeah, for this paper, what would constitute as a suicidal behavior for them to like quantify it, I guess?
Okay. I can't say that I've read the paper, but I did read, because when you talk about suicidal behavior, is talking about the psychological effects. Suicidal behavior. It's a good question. Maybe I can just get a paper. I'll get back to that. Okay. What constitutes as suicidal behavior? Because let me see. Because I've downloaded the paper. It's in my other laptop. I'll get back to you on that one. Yeah. Okay.
Okay. Thank you.
Right. Okay. Right. So summary effects. So it says summary effect was small, right? Really significant. So heterogeneity between populations. No time trends. Time trends meaning if the particular, especially when you're talking about age, you know, one study is using different age, you know, so there's no time trends. And effects were easily swayed and were largely dependent on individual studies. There's also a lot of variation in how people can, you see, when you talk about studies, there are statistics. And some people use different statistics, different important, different factors or parameters and so on. So that's why there's a lot of variation between studies, and then so on. Publication bias is whether what kind of study gets published, you know. So what my question here is that when you say there's a lot of variation, is it good news or bad news? Personally, I think it just says that it's not easy to determine, right? When you see something that there's so much variation, to me, it just implies that perhaps genetic factors are not that important, and perhaps, although there are, maybe the effect, as I said, effect sizes were small, and then the phenotype is G plus E. E is bigger. You know, perhaps the environmental influence is more significant, like, you know, how you behave, how you were treated when you're in your childhood, what do you have? Were you exposed to any particular trauma? You know, were you exposed to a lot of violence? You know, this kind of thing. So perhaps those factors plus you know, some genetic factors will actually lead to what we call as suicidal behaviors and things like that, right? Especially now when we start talking about, there's a lot of depression, right? There's a lot of students have been classed as depressed, and you know, so I'm not really. See, as I said, this is because I, in my personal opinion, I think there's a lot contributed by the environment rather than genetics alone. Okay, I think I should write that down. That you suicidal behavior. Okay. Okay, 10 more minutes. I'm finishing at paper number 20. So I've got, this is 17, so just a few more papers. Now, 17 looks at, looking at various studies, they realized that most, okay, here's the one that says, most cancer-associated SNPs variations are located outside protein coding regions. Yeah. So involvement of enhancer elements. When you talk about gene expression, you're talking about silencers, enhancers. These are elements. What are elements? Elements are region. A promoter is an element. Regions within the genome that functions as sites for protein binding. You know, so those are elements. Involvement of enhancer elements regulating gene expression and other yet unknown disease-specific regulatory. So I mentioned, so I say note, I mentioned this in paper 6. You know, when we talk about whether you look at genes or whether you look at non-coding. So this paper says, you know, most cancer-associated enzymes are actually located outside protein coding regions. Paper 18 is just general paper, looks at the value and issues in studying SNPs. 19, genetics and alcoholism. It says, this is interesting, you know, earlier studies show that there are variants in stress-related genes, right? So there are variants, but if this stress-related variants are only important if individuals are exposed to trauma, especially in early life. So this is what I'm saying, you know, like you have that predisposition, you have that, but if you're not exposed to trauma and so on, so you're fine. You may have some risks to diabetes, for example. But if you don't take sugar, you know, if you exercise well and so on, you may not get that. Not just diabetes, any particular disease. So that's why it says, you know, your phenotype is actually your genotype and environment. So instead of looking at causal variance, you may want to look at risk variance. And this is exactly what the genetic service companies do nowadays. It's part of your assignments later. Oh no, sorry, it's part of your exam question when I ask you to look at some of the sites and then comment. All right. So basically, earlier studies show that these variants are stress related and may only confer risk in individuals exposed to trauma. It is now recognized that genetic risks for alcoholism is likely due to common variants in very many genes. Right? Each of small effect. It's saying that, you know, there's a lot of factors contributing, but each of these are contributing very small effects. Although collectively, it may be significant. So there are rare variants with large effects may also play a role. So mentions the importance of gene interaction. Now, this has been going on for a while. Gene interaction. In fact, there was a time when people start questioning whether is there any such thing as single gene disorders. You know, when we do linkage studies, you know, pairwise, when we study dominance, you know, that part of the study, that when we're talking about Mendelian disorders, all of that specifically refers to single gene disorders. If you look at the Strachan and Reed book, Human Molecular Genetics, if you remember, I think it's chapter 14, I said we stop at 14.3. After that, you start talking about complex diseases. Complex diseases are like, you know, like diabetes, hypertension, high blood pressure, and so on. All this heart disease. So these are common. These are complex diseases. And previously, people cannot study complex diseases because it's complex. And the ability at that time is just to look at individual genes one at a time, and so on, candidate genes. But now with NGS, that's why there's a lot of focused study that looking at complex diseases, because for now you can look at the whole genome, right? And it's becoming more evident that genes don't work on their own. A gene function relies on other genes, and genes function in a network, right? Because that's why you have a lot of SNPs or a lot of variations that give you a particular phenotype. So unraveling that network is perhaps what people are doing, the focus of the research now, looking at function rather than individual things. Okay. Paper number 20. Oh, this is a paper. It says only 20% of smokers develop clinically relevant chronic. I'm not trying to promote anything, yeah, all right? And I didn't choose this paper specifically. I just choose the top 50 in my search and so on. It says, and why only 20% of smokers develop clinically relevant chronic obstructive pulmonary disease? Because genetics, right? So that means some people, like my grandfather, for example, if I remember my late grandfather, was you never see him without any cigarettes. You know, he's always smoking. You know, I remember when he came to the house, he was always asking my father to buy him cigarettes and so on. It was a long time ago, and he lived to about what, 70 or 80. You know, my late father never smoked. He lived to about 63. You know, so yeah, genetic is a factor. Okay, right. So we'll stop there for now. we'll continue in the next lecture on Tuesday. Hopefully we'll be able to finish the other 20 papers. But I think even at this point, you've already seen the strength. Oh, well, not just the strength, but also some issues. Because people can see a lot of variations. People can think, okay, these are various definitions, but they don't necessarily know how it works yet. And the fact that it also shows that, you know, while some studies are replicable, some studies can be replicated, some results can be replicated in different populations, others may not. So you really need to understand your disease. You need to understand your population. You know, so these are some of the issues that are pertinent in doing NGS studies. So I hope, well, at least the first 20 papers have given you an idea of what you can, what you cannot do, and realize the power as well as the challenges that lies ahead in some of the NGS technology. Okay,